Hi everyone. Today we're going to finish the topic of genetics. This is the third PowerPoint in genetics. It's a short one, but it was just a little bit too long to be able to add it on to the second one, so I made it a third part. So in this third part, we're going to address another issue in genetics. So far, we've talked about how autosomal traits are inherited, and those are genes that are on autosomal chromosomes. And we talked about dominant and recessive alleles and diseases associated with either a dominant autosomal gene or a recessive autosomal gene. Then we switched to talking about sex-linked gene disorders associated with the X chromosome. And so those are the sex chromosomes, or the X and Y. Males only have one X, females have two X's, and that there are a number of disorders associated with the gene on the X chromosome. And those are all more common in males because they only have one copy. And so if it's defective, they are going to have the disorder, whereas females can be carriers and not have it. All of the things we talked about so far dealt with traits, inherited traits, that are due to dominant and recessive alleles. So that in order to have a recessive trait, you would have to have two recessive alleles. Like blue eyes is a recessive trait. You can only have blue eyes if you have two blue alleles. If you have one brown and one blue, then you have brown eyes. Now we're going to talk about some exceptions to the rules about dominant and recessive. So not all traits are inherited as being either dominant and recessive. And so their inheritance pattern is a little bit different. So what we're going to talk about now are some of the exceptions to the dominant recessive inheritance pattern. The first one we're going to talk about is autosomal codominance. So codominance refers to traits where there is not just a recessive and a dominant allele. So like if you had a big R and a little r, but with codominant traits, both alleles are equally expressed. So that's a little bit hard to understand. I'll give you some examples that will help you understand the idea of it. So that, and since they're codominant, you don't have like a big and a little letter. That should be an R. You would have like two big letters, but different letters. And I'll give you some examples of that. So we're going to talk about a couple traits that are inherited by codominant genes. So it is not just a dominant and recessive, but that they're both there. And the two things we're going to talk about are sickle cell anemia, which is an example of this, and the human blood groups are also examples of codominant alleles. So we're going to talk about each of those. First, we're going to talk about sickle cell anemia. So sickle cell anemia is a very serious inherited disorder. It's associated with an abnormal hemoglobin being made. So hemoglobin is the protein inside your red blood cells. So here's a red blood cell here. And this protein inside your red blood cells is what transports oxygen. So inside every one of your red blood cells, you have thousands of molecules of hemoglobin. And when your blood goes to the lung, it picks up oxygen and then it transports it to every cell of your body. So it's a very important protein, and you need it to be able to get oxygen to all of the tissues of your body. In sickle cell anemia, there is an abnormality in the shape of the hemoglobin molecule. And because of its abnormal shape, it forms abnormally shaped red blood cells. And so this is an example here at the bottom. So instead of being kind of this ovally red blood cell, they kind of have a shape like this, which kind of looks like a sickle, which is used in farming. And so that's why it's called sickle cell anemia. And the problems associated with sickle cell anemia are due to this abnormally shaped red blood cell. So you have problems in that red blood cell traveling through your circulatory system. It plugs up capillaries, and it can get destroyed in the spleen. It also decreases oxygen delivery, and your body kind of recognizes that as an abnormal cell and breaks it down. And so there are lots of problems associated with sickle cell anemia, with this abnormal hemoglobin. So the gene for sickle cell anemia is the hemoglobin gene. So there is, let's say this is the normal. I'm going to change back to red. This is the normal hemoglobin gene. So a person who does not have sickle cell anemia would have two normal alleles. Then there is a gene for sickle cell anemia. I'm going to give it a little S here. So if in order to have sickle cell anemia, you have to have two of the S alleles. So it is kind of like a recessive disorder in that in order to have sickle cell anemia and to have symptoms of sickle cell anemia, you have to have two abnormal hemoglobin genes. But then people that are heterozygotes, so that they have one normal, and oops, I didn't want to write that there. They have one normal and one sickle cell gene. And so if you were to look at their blood, some of the red blood cells would be abnormal and some would be normal, so that they do show both. So that's why it's thought to be co-dominant, because you have both normal red blood cells and some abnormal red blood cells. And this condition in certain parts of the world is actually quite common to be a heterozygote for sickle cell anemia, because if you have sickle cell anemia, if you have the sickle cell gene, you are resistant to malaria. And worldwide, malaria is a major cause of death. It's not as significant in this country, but in many parts of the world, particularly tropical areas, malaria is a serious infection that can cause death. It's a primary cause of death in young children. And what happens is the malaria is caused by a parasite that is transmitted by the bite of a mosquito. And so if the mosquito carries the parasite and it bites you, then you can get malaria. And so parts of the world where there are lots of mosquitoes, so tropical areas, there is more malaria. But what happens if you have, let me backtrack, the malaria parasite infects red blood cells, and it doesn't like the sickle red blood cells. And so if you have sickle cell anemia, you're not going to get malaria, but you have sickle cell anemia, which is also life-threatening. But if you're a heterozygote, that means you have one normal and one sickle, you are resistant to malaria. You're much less likely to get it. So there is an advantage to being a carrier of the malaria gene, of the sickle cell gene. Whereas individuals that have two normal hemoglobins, they do not have sickle cell anemia, but they're also susceptible to malaria. So in areas of the world where malaria is endemic in the population, the occurrence of sickle cell, the heterozygotes, is much more common than it is in other areas of the world. If we were in class, I would show you a video about it, but I'm not sure that it would work here. So there is a link to it in the, I think I took the link out even because I wasn't sure if it would work in the PowerPoint for you. So that's sickle cell anemia. So it's one example of codominance, because if you have one gene for sickle cell and one normal gene, you have both normal hemoglobin and sickle cell hemoglobin. So that's why it's called codominance. The other example of codominance that we're going to talk about are the ABO blood groups. So the ABO blood groups are when you have your blood type done, usually they'll tell you something like you're O positive. And O is the ABO blood group, and that is inherited. And there are a couple different alleles. The plus or negative, or positive or negative, is a different gene for a different protein. I'll talk about that a little bit later, but they're inherited separately, so they're not connected to one another. So first we're going to talk about the ABO one. So ABO represents proteins on red blood cell membranes. And if you have an A allele, so everybody has two alleles. So you could be like, you could have an A, a B, or an O. So besides showing codominance, the ABO blood types also show something called multiple alleles, because every person only has two, but there are three different ones you could have. So everybody only has two, either an A, 1B, and 1O, but there are three possible ones. So like before when we talked about blue and brown, there were only two alleles. Here there are more than two alleles. A and B are codominant, and O is recessive to A and B. So what that means is, well, I'll get into what that means. They represent proteins on the red blood cell. So if you have an A allele, you have one version of the protein, the A protein, the A version. If you have a B allele, you have the B variation of that protein. If you have the O allele, that represents neither of those. If you, however, have one A and one B, because they're codominant, you have both an A and a B. If you have one A and one O, you're type A blood, because the O is recessive. So let's give you an example. If you're type A blood, you can have either two A alleles or an A and an O allele. If you're type B blood, you either have two B's or a B and an O. If you're type AB, and that's actually the rarest blood type, you have one A and one B allele. And if you're type O blood, you have two O alleles, and they're the two recessive alleles. This is also a good example because we have a tendency to think because something's recessive, it's rare. But type O blood is actually the most common. So even though it's recessive to A and B, it's still the most common allele in the human population. The significance of this is if you have type A blood, for example, and you get a blood transfusion with a different type, say you get a blood transfusion with type B blood. The B blood has these B antigens on them. And when that enters into your bloodstream, your immune system is kind of checking for things that aren't supposed to be there. And when it sees a B antigen, it says, whoa, that's not supposed to be here. And so it will destroy it. So that if you have type A blood, you cannot get a blood transfusion with type B blood because your immune system will attack those cells. Likewise, if you have type B blood, you can't get a blood transfusion with type A blood because your immune cells will not recognize those blood cells. If you have type O blood, you can only get a blood transfusion with type O blood because your immune system would recognize neither A or B. However, if you have type O blood, you can give blood to any other person because it has no markers for the immune system to reject. So if you have type A blood, you can get blood transfusion with either type A or type O. If you have type B blood, you can get transfused with either type B or type O. If you have AB, you can only get AB or O. And if you have O blood, you can only get type O blood. O is considered the universal donor because it can give blood to any of the other blood types. Oops, that should be donor, not blood. Sorry. And AB is the universal recipient because they can get blood from A, B, AB, or O. So they can get it from any of the other blood types. So O can give blood to anyone. AB can get blood from anyone. And so that's the way that those blood groups work. And so they're inherited. And so you can do a Punnett square. So if one parent is type AB, they would give half of their sperm or egg would have the A, half would have B. If the other one is O, then the possible outcomes for the offspring would be AA, which is type A, AB, which is AB, AO, which is type A, or BO, which is type B blood. So with this cross, there's no possibility of having a child with type O blood. In this one, where you have an AO and a BO, you have a 25% chance of having type O, a 25% A, a 25% B, and a 25% AB. So there's one of every blood type. And this just shows you the distribution within the population. The average US blood population, almost half of the people are type O, then A, and you can see AB is the rarest. And that varies with certain groups. Native South Americans, there's only type O. The two AB variations do not occur. The other component of blood typing, which isn't an example of codominance, but I'll talk about it here since we're talking about, is the positive or negative. And that actually refers to something called the RH factor. And RH stands for rhesus monkeys because this was discovered in the blood of rhesus monkeys, but we have the same thing in our blood. And you can see almost a very large proportion of the population is Rh positive. So Rh negative is much more rare. In the US population, only about 15% of the population is Rh negative. And the Rh factor refers to another protein on the red blood cell membrane. It's different than the ABO ones, and it's inherited separately by a different gene. But you either have that protein or you don't have it. So if you have it, you're Rh positive, and if you don't have it, you're Rh negative. The problem for this only occurs if you have an Rh negative mom. So an Rh negative mom does not have, she has two alleles and both of them do not have the protein. So she's Rh negative. And if the father of her child is either a plus plus, has two positive alleles, or a heterozygote has one positive and one negative, then she has the possibility of having an Rh positive child. So it can either be Rh positive or Rh negative. The problem is if the mom is Rh negative and the fetus is Rh positive. And it's usually not a problem for the first child. What happens is the fetus is producing this Rh positive blood cells, but there's not a lot of mixing between maternal and fetal blood until delivery. So when that fetus is delivered, the mom gets exposed to the Rh positive and makes antibodies to it. So if there's a second pregnancy with a Rh positive fetus, then you can have problems with the mother's immune system, causing problems for the survival of that fetus. Oh, let me just pause there. And that can be treated with medication that prevents the immune system from recognizing that Rh positive protein. And so that's something routinely tested for when a woman is pregnant and has gynecological care with the obstetrician. They would do a blood type just to see if the mother should happen to be Rh negative and the father Rh positive, that they would then treat. There's a drug called Rogam that can be administered to prevent that immune response from happening and to prevent any problems with the pregnancy. So far, all of the traits that we talked about were involved with variations in a single gene. So, and where you have different alleles for a single gene. Many of our characteristics or traits, and remember traits mean kind of your characteristics, are not caused by one gene, but by many genes contributing, and then also some environmental influences. So things like height. There's not like one gene and you're either going to be tall, short, or if you're a heterozygote, maybe in the middle. So there are many things that affect your height. Besides there's genetic components, there's hormones, there's diet. So it's not a single gene trait. So these things are called polygenic because there's many genes that will influence that. Skin color. There's not just two skin colors, hair color. There are even eye color, which we talked about brown and blue, but there are other genes that influence eye color that leads to green eyes and hazel eyes. So those are all called polygenic traits, and many of our traits are polygenic. And in polygenic traits, you don't get just brown or blue or two possible. You get a whole range of possible phenotypes. So here we have height. So when you were to take a population and graph their height, you don't get just two different heights. You get a whole range of heights where the average in the middle would be the most common, but they're kind of spread out each way. And that's kind of characteristic of polygenic traits, where you get a whole range of possible phenotypes. And so that is, many traits are controlled by multiple genes, not just a single gene. Okay, I think that ends the discussion on genetics. So I will post a Genetics 3 quiz, and then I've decided to make exam 4 on the material of meiosis and genetics. And so I will go ahead and put up exam 4 also that you can take at this time. Bye-bye.